Everyone, thank you for joining us. I really want to extend a great welcome to Jennifer Rawlings, who's in our very own Columbia, South Carolina. So welcome to Columbia Scuba's webinar series that goes along with our Dive with Purpose theme. And we try to do these monthly to give people an opportunity to hear more about very interesting topics that are near and dear to our hearts and the hearts of all divers, really. And that's how to protect our environment and how to become a part of any programs that are out there that we can learn more about and support. And so tonight we've got Jennifer Rawlings with us. Jennifer is at Riverbanks Zoo in Columbia, South Carolina. And Jennifer, in the brief time that we've had to talk, Jennifer says that she really developed a love for the ocean at an early age. And that love for the ocean had prompted her to seek her undergraduate degree in marine science. And during that period of time when she was in college majoring in marine science, she had an opportunity to work at a public aquarium. And in that time, she really developed an affinity for taking care of the animals and caring for all the things and parts of the zoo. And the animals that she was in charge of really had a place in her heart, which is awesome because of our love for marine animals. Jennifer is in charge of the, and I guess you lead a team, Jennifer, of folks that are helping protect and manage a bunch of coral that's being placed at Riverbanks Zoo, which is what we're trying to learn more about tonight, as part of the you called them, I think you called them refugee coral, and that they're here from Florida as part of the Florida Reef Tract Coral Protection Program. And so Jennifer, thank you for being here. And I will mute myself now and I will turn it over to you. We look forward to hearing from you and learning more about what y'all are doing at Riverbanks with the Reef Tract program. Thank you so much.
As Lawrence said, I'm Jennifer Rawlings, and I am really pleased to be here tonight talking to you about the Florida Reef Tract Rescue Program and our role in it at Riverbanks, and also how you guys can help or get involved. Okay, so this is kind of an outline for what I'm going to be talking about tonight. We're going to be looking at the disease, so I'll explain a little bit more about what it is, the current response to this threat, Riverbanks' role specifically, and how you can also play a part in reef recovery efforts. So the disease is called stony coral tissue loss disease, and it was discovered in 2014 off of Miami-Dade County in Florida. And it was a new disease. They hadn't seen anything like it. It's been spreading rapidly throughout Florida and also the Caribbean in the last few years. You'll also hear folks refer to it as SCTLD, which is just the acronym for stony coral tissue loss disease. So this is the map that's been produced that shows how the disease has spread since 2014. And you'll see it started spreading north and then south along the reef tract in 2015, all the way down through the lower Keys. And sadly, in May of this year, scientists actually discovered SCTLD in the dry tortugas. So it has gone completely through the entire reef tract now. I don't have that on this slide here, but yeah, it has gone through the whole entire tract now.
Jennifer, you said that they've pinpointed it from starting in Miami-Dade area?
Correct. Yep.
And is there an event or some specific thing? What do they think? Where did it come from?
So there is some speculation that there was some dredging in the area and those types of activities that could have started some kind of flare up with this disease. Scientists, and I might mention it later, they still haven't exactly pinpointed what this is. So they've been looking at etiologies of bacterial or viral etiologies, and they haven't really nailed it down yet. I actually sat in on a presentation from some of the folks that are doing that work, and one of the things that they're looking at right now is the zooxanthellae, so the algae that live in the coral tissues. It might actually be a disease of the zooxanthellae, and when they become compromised, it actually causes the disease in the coral itself. So, but yeah, I think they're thinking some dredging activity possibly could have been the start of it. And then, of course, coral reefs are so compromised because of other activities, climate change, ocean warming, that it's kind of a perfect storm. All right, so why so much attention on this particular disease? There are many coral diseases that affect reef building corals, so why so much attention on this one? And there's a couple of key factors. So with this outbreak, there are many species that are affected. They found over 20 species that are susceptible to this disease, and there are only about 45 species in the Florida Reef Tract. So nearly half of the species have been known to be affected by SCTLD. There is a high prevalence. So what that means is that out of every 100 corals surveyed, that 66 to 100 of them have this disease. So it's it's highly prevalent in the population. There's a rapid mortality. So once a coral becomes sick, it can completely die within weeks to months. And that's a very fast progression for disease. There is also a high transmission rate. When it's found on a reef, it can spread rapidly across the species that are susceptible. It has a large geographic range. So as I mentioned, it's spread across the Florida Reef Tract, but now it's also down in the Caribbean. And it's covered over 96,000 acres since it was first discovered in 2014. So a huge swath of reef, basically. And the long duration, again, since 2014, most coral diseases are very punctuated. They might happen for a brief time, or during the summer, there might be this cyclical disease pattern when the ocean temps warm up that corals become ill. But we're not seeing that cyclic nature or stops and starts with SCTLD. It's been consistently spreading and decimating the reefs for 6 years. So long duration. So this is an image I think is pretty stark, and that's why I'm sharing it. You can see in the course of approximately a month what SCTLD can do to a single coral colony. So this is about a 60% death in this colony, and you can imagine that happening across an entire reef system. It can be really devastating, and folks have talked about it as an ecological crisis, and I think that's true. Seeing photos like this and imagining reefs like that, it is a crisis. So what is the response then? Clearly there needed to be some kind of response to what was going on once managers and scientists realized this wasn't going away. They knew it was a disease unlike any other, and that a large coordinated response of some kind was going to be necessary. So this graphic just shows that there are multiple teams that are working on this problem. You have scientists in the field who are monitoring where the disease is showing up and how it's affecting corals and the spread. And then you've got pathologists in the lab who are, again, looking under the microscope to try to determine what's happening at a cellular level. So all these teams are working together. It's really, really an amazing effort. And where Riverbanks comes in is on the coral rescue team. So that's our little pocket that we're working in. And so what is the rescue team? So in late 2018, the partners who were already working in Florida on the disease response reached out to the Association of Zoos and Aquariums, also known as AZA, to ask for help. And AZA members have expertise and experience in caring for animals. And we are so proud that we were able, this is Riverbanks Zoo, to step up quickly and offer our assistance. So the rescue team consists of AZA members as well as other coral care facilities with expertise in staging and caring for corals. So what has been our role as this has evolved? We were fortunate enough to be one of the kind of early adopters to the rescue program. The original purpose of the project was to bring unaffected corals from ahead of the disease front into land-based holding facilities in order to maintain genetic diversity of susceptible species. So those 20+ species, those are the susceptible ones, and those are the ones that are in the program and that folks like Riverbanks and other facilities are caring for. In other words, you know, the corals are dying on the reefs. Can we save them by keeping them away from the disease, by keeping them in land-based systems? So once Riverbanks was approved to be a holding facility, we immediately began setting up a dedicated aquarium system in early 2019. We had to identify an area that was separate from our main collection of corals due to biosecurity requirements. So you can see here our aquarium supervisor setting up our systems. And this is just a shot of the systems when we first put water in them, which is pretty cool. See if I can get this video to play. So it's just running water, but that's the thing that excites aquarists. And then we received our live rock, which you can see in that right-hand photo, just to start the tank water cycling so that it was ready to accept corals.
Now you say live rock.
Mm-hmm.
Is that just like limestone from the reef that's taken in and put in the tank?
Yes, exactly. So in this case, everything that we're receiving is vetted through FWC, so Florida Fish and Wildlife. And so this was actually rock that they collected, and it has organisms growing on it, little critters that basically kind of help seed the biological filter in the tank. And so that helps make a healthier environment for when we introduce the corals. It's a very, very typical way of managing aquarium systems. Okay, there we go. So this is a shot of when our corals actually arrived. This was in April of 2019. And we were the 4th facility outside of Florida to receive corals into our care. All of our corals were collected from one site, Lou Key, and this was ahead of the disease front at the time. So it hadn't come down into the lower Keys yet at that point. After collection, they were held at the Keys Marine Lab, which I know I sat in on a presentation earlier in this series that was great from Keys Marine Lab. And they held our corals and then they sent them on to Columbia. So that is a pretty cool connection. But in this photo, what's happening is the aquarists are acclimating, getting these corals ready to go in the tank. They can't just go from their shipping container to the tank. And you can see that they're shipped in cardboard boxes with bags and packaging for just keeping them safe on transit. And they just come FedEx to us, basically. This next slide is looking down into one of those boxes. So this is how the corals arrived. They were shipped in these plastic baskets. And we had a few corals at a time per basket. And these particular corals actually came off of their tiles that they were mounted to. They came off of those tiles during shipping. But each coral is mounted to a tile, and each tile has a tag that is associated with that coral. So that is how we track each individual colony, is with these tags.
Just curious, Jennifer, if I may, when it's being shipped, was there anything in there to circulate the water, aerate the water? It just seawater and coral. That was it?
Yes, and they will pack them in a particular way. So with corals, what they generally will do is fill the bag probably about halfway with water and then seal it so that there's no extra air in there so that it doesn't have a chance to become turbid or full of air in the water. We want to not introduce extra air because it can agitate the coral tissue. Yeah, but yeah, it's very basic and they seem to tolerate it very, very well.
Could I ask a question about the duration? Like how many hours or how soon does it need to go from there to you?
Sure. So they come priority overnight, which means they're packed in the afternoon. Like in our instance, Keys Marine Lab would have packed them up in the afternoon. They would have traveled overnight to us and we received them the next day. So it's probably not more than a 24-hour transit.
Okay, thank you.
You're welcome. And as an aside, there's a really great tool online. It's a cumbersome URL. I've listed it on this slide, but you could probably Google Coral Rescue Coral Monitoring Dashboard and it would probably come up. But this is a really, really cool tool if you want to see current information about what's going on with the rescue. So this dashboard provides information about the species rescued and the facilities that are housing them. I've sorted the data on this slide for Riverbanks corals. So you can see that we're currently caring for 44 coral colonies. And the red dot on the map at the top indicates Looe Key, which was the collection site for all of the corals under our care. So our job now is to simply provide the best care for these corals that are serving essentially as a living gene bank for their species. And this is just an image of what the systems look like now. We actually had to add that third one on the left side because we've had so much growth in our corals that we didn't have enough space for them, which is a good problem to have. So this is just a video clip of what the space looks like. We do have some automated systems, which is really, really nice for us. And this is tucked away in a corner due to biosecurity guidelines that we have to follow.
Jennifer, if I'm getting ahead of you, and I apologize, but in the way that you guys monitor the coral that you're keeping right now, is there any ongoing comparison between coral being kept in other repositories as far as growth rate and health and even compared to what's dying off in the Keys right now?
We, as far as I know, aren't being directly compared to what's going on in the wild, so I can't really speak to that end of it. The Florida Fish and Wildlife may be doing those comparisons, but I don't know. But I know for the coral holders, so facilities like us and the others, I'm really glad you brought that up because I don't address it in my presentation, but we have regular reporting we have to do. So at this point where we've been holding corals for a couple of years, we have monitoring photos and measurements that we submit every 6 months. When we were in our first year of holding, we actually had to do that monthly. And so they've been able to track the growth rate of all of these corals at Riverbanks and at the other facilities from the time they arrived all the way up to the current time. And we have had some really amazing growth rates, which is incredible. And the Florida Fish and Wildlife is very excited to see how the corals are doing in our care. We're learning as we go. They're learning about these corals. We actually had a group call today. So we meet once a week to discuss what's going on in our tanks and discuss water quality and all kinds of topics. And today we did they call it a deep dive, which is just looking a little bit more into each individual species. And we learned about the Euphasmilia, which is smooth flower coral. I'm sorry, I don't know all the common names. Smooth flower coral. And we're talking how in our care, we see that they're very, the coral polyps are very fleshy and swollen, and they look really just fat, is what we were saying. They're very fat. And we felt like that was a sign of a healthy coral. And one of the Fish and Wildlife folks spoke up, and she said, you know, in the wild, we don't see corals that look like they do when we see photos that you guys are caring for, the corals you are caring for. She said, in the wild, they're always very thin and just not as puffy. They're not fat. And she said, you know, it's a testament to the great care they're receiving. I am going to talk about feeding. And we feed them a lot, and it shows. They're really robust and happy. So yeah, we're always chatting and sharing information, and it's an important part of what we do, is to learn from each other what is happening in our systems. Here's another just clip, what some of the corals look like. And we do have a number of pumps that recirculate the water. You'll see there's some snails in there. So we have janitorial animals that help keep our tanks clean, in addition to us hand scrubbing things and picking algae and all those kind of fun things. So I think these corals are just amazingly beautiful. I hadn't seen Florida corals up close like this before. And they are just amazing. So I wanted to share some photos of them so you guys could see them as well up close. Most of the corals in the rescue program have not been maintained long-term in human care. So as I said, we are really learning as we go. And figuring out how to feed these corals has been really interesting. So that Eusmilia fastigiata, that's the one on the right-hand side, also called EFAS. If you look on the tag there, that is how we often refer to them, is by this acronym. So that's our EFAS on the right-hand side. And they really like meaty pieces of food. As I mentioned, they're pretty hungry. They like the meaty, bigger pieces of food. And the guy on the left, who is Macidophilia aliciae, or Molly, that's easier to say, like smaller pieces of food. You can see he doesn't have the big polyps like the EFAS. He likes smaller food. So we had to figure all this out as we were learning how to care for these corals. And this is what the different foods look like. So on the left-hand side is that bigger, meatier food that the EFAS would like. And then on the right-hand side is kind of what we call a coral slurry. So it's very small plankton that we mix together and feed out to some of the guys that like the smaller food items. And really, we're just learning to accommodate different feeding preferences, and it's been pretty cool.
Are you growing your own plankton and whatnot, or do you get it shipped in? How do you actually maintain the food supply?
Yeah, great question. In this case, we can actually use prepared products. So there are companies out there that will aquaculture or sometimes wild harvest, but they will get the plankton and they'll freeze dry it. And it's basically a prepared commercial product that we can then reconstitute or add in. We actually add in some additional basically vitamins. So there's also vitamins in the water, and then that's what we feed to the corals. We do culture at the zoo some plankton. We do green water, so algae, and then also artemia, which is just a brine shrimp. And but those are for some of our other animals. And actually, because of the biosecurity requirements with these corals, we wouldn't be allowed to feed any of those cultures to these corals, just because it could present a risk of cross-contamination. So maybe one day we'll set up our own cultures just for these guys. But the commercial preparations work really well.
Thank you.
Absolutely. So what we do to feed them is just turn off the wave maker pumps to make the water kind of still. And that is kind of serves as a cue for those corals to say, hey, this is feeding time. I know what this is like. And they respond by extending their feeding tentacles and opening their mouths, which is crazy to watch. And I don't know. See. Yeah, I don't know how to. Oh, wait. Nope. Okay. Well, I can't find my pointer, but maybe I can. I don't know. Anyways, yeah, so they've got their feeding tentacles extended, and they're ready to feed here. Oh, don't know why it's not advancing.
So
Sorry,
do you simulate daylight and nighttime UV and different lighting levels to cause them to come out?
We do. Yeah. So, and actually we don't, at Riverbanks, we don't go as far as some of the other facilities do. So for instance, there's a facility in Florida that they actually come in and do feedings at night in the dark. So they will have red light headlamps on, and they'll feed the corals at night when they're just naturally, some of them will naturally be feeding at night. We have conditioned ours to feed more in the daytime, and they seem to tolerate that fine. It doesn't upset their biological clocks. But one of the cool kind of innovations in reef animal caretaking over the last few years is really the advent of LED lighting. And it's been really great because it is programmable. So we can dial in the exact color we want. If we want to mimic, high noon sunlight, we can do that. If we want to mimic a cloudy day, we can set up a program that will do that. If we want to ramp up intensity, going from 8 a.m. to noon and then ramp down from noon to 4, we can program that in and the lights will automatically do that. So those are all things that we do to mimic what would be going on in the wild, which is really cool. Now, why my slide is not advancing here? There it goes. Okay, so the coral on the left, I don't know if you can kind of see that little puckery part. That's actually the mouth open and ready to feed. So that big fleshy part is one polyp, and the mouth is right in the middle and is ready to feed. This is a Musa angulosa, what we call a mang. And then the technique is simply using a pipette to just pulse that food right to the corals. And of course, these guys are reef building corals. So as I mentioned, they've got that bright lighting, which also provides them with energy. So the feeding is supplemental, but we have found that it is essential with these Florida corals. We've been maintaining corals at Riverbanks for decades, but never Florida corals. So they've always been Indo-Pacific corals, and we don't feed those guys. They don't need it. They get plenty of energy from the lighting in their tanks and probably the occasional fish feces, putting nutrients into the water. And they do fine without supplemental feedings, but these Florida corals really, really need the extra food. So that's been just a really cool thing to learn about and figure out. So one of the other really cool experiences that we've had with these corals is the formation of gemma. So on this coral, you can see those really fleshy parts kind of in the forefront of the screen there. And those are the gemma. So the rescue program is developing a strategy for sexual reproduction of some of these species of coral. But in the meantime, some facilities, including us at Riverbanks, have seen the formation of these gemma. their type of asexual reproduction, essentially what is a clone, in which the parent colony starts to form extra tissue in a small area. So that's what you're seeing there. The tissue grows and eventually forms a small internal skeleton and then drops off of the parent colony. In nature, this rolling stone, as they are sometimes known, would be moved around by way of action until it settled on another area of the reef. So that's an easy way for a coral to asexually reproduce and then spread on the reef. So this is a diploria, a D-lab. This is a Colpophilia natans, we also call CNAT. So this is a parent colony. This is the gemma that was produced by that parent colony. This is what it looked like shortly after it dropped off of the parent colony. And the white discoloration on the tile is actually super glue. So that's how we adhere corals to tiles when they're this small, is with super glue. When they're bigger, we can use epoxy, but the super glue is a little bit easier when it's small guys like this. So this is what that same gemma looked like after about 8 months of growing.
Wow.
Yeah.
Is that a 4x4 tile?
It is.
Okay.
Yep. And then this is what it looked like another 8 months later. So
Beautiful.
I know. And you can see, I mean, it's just taking up a little section of the tile here. And then right there, it's almost right to the edges. So it's truly amazing. And we have a couple of species that we've gotten gemma from, which is exciting. So where are we now? We are continuing to hold these corals and we're looking to the future. So our original commitment to the rescue program was for 3 years. And none of us on the rescue team really knew what we were doing, as such a large collaborative effort had never been undertaken before. We now have a better understanding of how each individual part is coming together to make the whole, much like individual coral polyps forming a colony. Riverbanks Zoo, I'm excited to say, has committed to an additional 3 years, and we now have an opportunity to bring our back of house coral tanks up front for our guests. You may know that our aquarium building closed in August for an extensive renovation. One of the exciting parts of this project is the construction of a dedicated coral lab to house these valuable corals. We'll be expanding our holding space to allow for additional specimens at Riverbanks or to help meet whatever needs there might be as the rescue program progresses. So we are very, very excited about this. We actually just started demolition on Monday. So yeah, great stuff going on.
That's awesome. With this lab that you're building,
Mhm.
Jennifer, do you guys have an active internship program with maybe the marine science programs at the university?
I love that question. Yes, we do have an active internship program, and we do work with USC through the marine science program to recruit marine science students specifically. But we always have an open application, and anyone is welcome to apply if they have an interest. And I think we're actually going to be looking for interns or volunteers who have experience working with corals. Once this lab comes online, I think we're going to need extra help and hands. So we may be recruiting specifically for aquarium caretakers.
That's pretty awesome. For experience, you know, maybe it's more extensive than the folks that in our dive family have had, but it would be someone who's like taken coral courses, done hands-on cleaning and stuff like that, or what kind of experience are you talking about?
That would be great. You know, it could be at a very small level, someone who has a reef tank at home. They would be a valuable asset. They know, you know, the basics, and we would train them on the specifics for our corals. Or anyone who has an interest really, and can, you know, learn and take direction and wants to help. We're always looking for folks that have that energy and excitement, even if they don't have all the experience. But yeah, I think students that have a particular interest in coral science or marine science would really benefit from working with our corals. And I will mention as well, we also do have a diving volunteer program. It is not active right now, just because of COVID issues, as I'm sure everyone's aware. But that is something to look for if you're interested in diving at the zoo in the future. We post those online and are always looking for great volunteers to help us clean the exhibits and feed the animals. So. All right, so there are ways that you guys can help with this effort, not only for the rescue program, but for corals in the wild. And, you know, this program for us has helped. I feel like it gives hope for the future of the Florida Reef. You know, we're doing active hands-on things and just excited to be working in this sphere. So my first thing would be to encourage you to come see our new coral lab when it opens. Come see these guys in person. You know, support the zoo and the work we're doing. And it's not just for corals, but it's other wildlife and other wild places. So that is a plug for us, shameless, but yeah, I'm proud of the work we're doing. Let's see.
Jennifer,
Yes.
do you have a estimated date of when that'll be open?
We're probably going to be closed for a full year. So I'm anticipating that we will reopen to the public, you know, next August or September.
Okay, thank you.
You're welcome. Okay, I'm going to see if this will play. This is a link to a little YouTube video. That is not playing for you guys, is it?
Don't see it playing yet.
Okay. That may not work. I can't remember what we actually did to get it to work. It's just a little 30-second clip. But basically talks about, you know, just when you're out diving or on the beach, is to just be mindful of, you know, where your trash is going. Plastics are a big problem in the ocean. So I'm always encouraging folks to use alternatives if you can. Think about the sunscreen you're using. You've probably heard of reef safe sunscreen, and, you know, just be mindful that sometimes the sunscreen you choose can put chemicals in the water. So, you know, to just be aware of that. Yeah, that's just the 30-second clip. So I am sorry I could not get that to work. Okay, let's see. This is a great infographic that I pulled from some Florida website. I can't remember, might have been Florida Department of Environmental Protection. But, you know, just again, to be mindful as a diver, you have a great opportunity to help the reefs by disinfecting your gear between dive sites, especially with SCTLD. You know, we're not quite clear on the vectors of how it moves. Dive gear could be a potential vector. And I would say, you know, if you're working with an operator and you don't see a disinfection or, you know, a dunk system where you're cleaning gear off in between sites, speak up. I think that's, you know, a great opportunity to say, hey, I know about this disease, you know, and is there a way we could do this better? So, yeah, I like this infographic a lot. And then, of course, I love encouraging folks to participate in citizen science. As divers, you can provide boots on the ground information about the status of the corals that you're seeing, both healthy and unhealthy, when you're on your dives. There are several conservation groups that have online reporting for observations made throughout Florida and the Caribbean. And I know that Columbia Scuba has wonderful active participation in coral outplanting efforts, which is also great citizen science, and contributing, again, to boots on the ground reef conservation efforts. So I would encourage you to seek those out, read up on how to do reporting before you go on your next trip. And you can contribute directly to science, which is awesome. And then finally, I would encourage you to be a coral champion. So talk about the issues of ocean conservation with your friends and families. I mean, you guys are key players. You see in situ what's going on the reefs, and you love the reefs and the animal life, and you have that passion. And so encourage those that you know to do what they can from home. Share with them what you know and have learned. Simple things like having them use a reusable water bottle instead of a one-time use plastic bottle. Simple things like that. But yeah, think about that. Be a coral champion. And then this is just my last slide. These are some links for further resources if you're interested in learning more about SCTLD, the disease and the response, and then how you can help. And that last link is a really great story that tells more about AZA, the involvement of facilities like Riverbanks, and how we're working together to give hope to the Florida Reef Tract. So that is it. And I'm so happy to have shared with you guys, and I would love to hear any questions you might have.
Jennifer, first, I want to say just thank you so much. This has been incredibly informative and really, really awesome information about the things that you guys are doing. And it's really nice to hear about the success you're having with the growth rate and the health of the coral. I'm kind of excited about it. It's
Good.
It's great information. And I think what I've done is I've actually taken photographs of the links for the resources. And so
Great.
we'll include that with other mailings that we send out. So I really appreciate it. Going back to a question that Kelly had typed me in the screen earlier. So you had said that this thing was first identified around 2014.
Mm-hmm.
And so just in the course of 6-7 years, it's expanded throughout the Keys, all the way down to the dry Tortugas now, and all the way up to upper like Jacksonville area, or just
Yeah.
about, because I know there's not a lot of coral up there, but
No, let me see if I can get back to the little map. Yeah, all the way to the northern reaches of the reef. And sadly, I don't have a map here, but again, you could check it out online. It is quite prevalent in the Caribbean as well now.
do you know if this has actually been found? Oh, so it's just up above Palm Beach, Martin County. Okay. That's pretty much where the reef ends.
Yeah, that is, yeah.
Okay.
So when you were, talking about the Caribbean, that was kind of my question as well. Like, is it in the Bahamas, or we're seeing it more south?
It's more south. So, there are some water circulation patterns that I think that they probably think that is one way that the disease is conveying throughout the reefs. But yeah, I've seen it further west and then also south.
Okay, so like Bimini would be involved. It's closer to the Florida coast. Okay.
Yeah.
Has this been spotted in other oceans, in other countries?
No.
Wow.
And, I hope it doesn't, go any further, or something like it doesn't creep up in other areas. But it is still contained and only been seen in this region. Yeah.
I noticed that it looks a lot like, from the pictures that you've shown, it looks like it's being bleached. It looks like coral bleaching so much. So is that why they think it's a disease of the zooxanthellae? Because from what I understand about bleaching, I'm not a coral expert, but I know that like it kind of causes like that, at least when you're bleaching, it kind of causes the algae to abandon the coral, right? Like it.
Exactly. Yeah. So bleaching is when a coral expels its zooxanthellae. So it has no more of that algae in its tissues. And so the algae also gives some of the color to the coral. So that's why bleached corals look white. But the tissue is still there. So it's still alive. It still has a chance to recover. Sometimes those zooxanthellae can recolonize and the coral can recover. With SCTLD, it's actually complete tissue loss. So that is the white on the picture here is bare skeleton, and they can't recover from that. So
Have they tried any type of like an antibiotic or some kind of a salve or something to see what could possibly help regrow?
yeah, they have done trials with different types of treatments. Early on, one of the things they tried was actually a dental paste that was impregnated with bleach. The bleach didn't work so well. It wasn't stopping the progression of the disease. So then they did try some using that same kind of dental paste, but with antibiotics, and that has actually seemed to work very well.
What particular type are they kind of going with?
I'm sorry?
What particular type of an antibiotic are they?
Amoxicillin.
Oh, wow.
Mm-hmm. And it works. It's labor intensive to apply because you would have to go to each coral that's sick. So, they're still looking at other avenues, and they aren't quite sure why an antibiotic is working. They're not sure that it's a bacteria that it's working on, but it's just very strange, the whole thing. And how is the zooxanthellae involved? Is that a primary cause or a secondary thing going on? And so scientists are still trying to work all that out.
Wow.
Just start from that particular photograph of less than a month.
Yeah.
It's gone from 5% to 60%.
Yeah.
Insane. So
Any
sad.
other questions for Jennifer?
I was actually wondering, because you talked about like wanting to like preserve the genetic diversity of the reefs. Is there anything, I'm sure it's going on, somewhere, but is Riverbanks looking at potentially genetically sequencing some of the coral to kind of store the genetic information for future like sexual reproduction and outplanting efforts?
You guys have awesome questions. I love this. I'm serious. This is great. So as I mentioned, we have weekly calls with all of the group members, and on some of those calls, we go deep into particular species, and it's usually someone on the FWC team who is doing that presentation for us, and the last few times they have been able to share genetic data with us. So they took samples of all the corals when they were collected, and they've been doing genetic analysis, and what they're able to tell us is how related the corals that we're caring for are. So for example, on the call today, they were talking about the EFAS corals, and a lot of them, the majority of them were collected in the dry tortugas, but from different sites. And so they were able to see that a lot of those are actually siblings. So they're pretty closely related, but they're 2 miles apart in some cases. So, they're able to learn about how far these coral larvae can spread. I mean, 2 miles, that's a huge distance for a tiny little larvae to travel in the ocean and settle down and make a new colony. So they are slowly working through the genetics, and what that's going to do in the end, in addition to cool information like that, learning about what's going on in the wild and how corals are forming and spreading along the reef. But it will inform their sexual reproduction strategy. So they'll be able to pick the best parent colonies and shift them around between facilities, which we're very used to doing with animals, putting together ones that are going to make the best genetic combination for the offspring. So we can do that with corals as well. And they'll probably have us shifting different colonies around, and the facilities that are going to be working on the sexual propagation will be able to do that. And yeah, it's very, very cool stuff. But I'm glad you asked, because yeah, the FWC and their partners are doing that work. Yeah.
That's awesome. Thank you.
Jennifer, again, I really want to thank you so much for your time and your expertise and your passion that you've shared with us. You guys are doing great work. This is really cool.
Thank you.
And so we've been very excited to learn about what Riverbanks Zoo is doing to support the long-term health and well-being of our coral reefs down in Florida. So thank you so much for presenting this. I thought it was very interesting. So did a great job. So
Well, my pleasure, and thank you for inviting me. I had a really great time.
Thank you.
Thank you, guys.
hope you have a good evening. Thanks so much for joining us.
Thank you, Jennifer.
Bye, y'all.
Take care. And
That was cool.
very cool.